Hello, everybody, again. Welcome to our e-seminar series today with Professor Milica Radisic. So let me share my screen and share with you some housekeeping notes, and we start off from there. Okay, as always, we record this talk and we will post it on our YouTube channel. Please also take the polls that it's on the bottom of your page. This helps us to improve the quality of e-seminar series. We put outstanding list of speakers for our fall quarter. So please follow us on LinkedIn and other social media and our Twitter account to get the most updated schedule for the talks. And our next e-seminar will be with Professor Meg Griffith from University of Montreal. She will be talking about in situ tissue regeneration, a journey from bench to bedside and back. You can always follow our Twitter channel at BME and the organizers, Professor Akbari and myself, and our coordinator, Mr. Karamzadeh. We would like to thank our sponsors, Transmedtech Institute, for providing the resources and sponsorship to these seminars. Let me just remove the timing here. Yeah, so Transmedtech Institute aims to support the development of innovative medical technologies, train the next generation of professionals, and make innovation in life sciences and engineering a source of wealth for society. Based on a living lab approach, Transmedtech provides an integrated environment that supports interdisciplinary collaborative processes and co-creation of new medical technologies and interventions to catalyze their development and adoption by users. And our next sponsor is Fluidform. Fluidform has the exclusive license to fresh 3D bioprinting technology from Carnegie Mellon University, and is focused on advancing applications of the technology in biofabrication, regenerative medicine, and other industries. Life supports bioprinting support gel is used by researchers worldwide to significantly improve their ability to print cells and soft deformable biomaterials into complex structures with high fidelity. Fluidform has offices in Boston area and in Pittsburgh. So by that, I would, yes, hello, Professor Radisic.
Thank you very much, Human.
Thank you for connecting. Thank you so much for accepting our invitation. So I just need to unshare my screen. And then, so let me introduce Professor Radisic. She's a well-known scientist, pioneering scientist in cardiac tissue engineering, and my mentor and previous supervisor. Dr. Radisic is a professor at the University of Toronto, Canada Research Chair in Functional Cardiovascular Tissue Engineering, and a senior scientist at the Toronto General Research Institute. She's also the Associate Research Chair for the Department of Chemical Engineering and Applied Chemistry at the University of Toronto, Director of the NSER Create Training Program in Organ-on-a-Chip Engineering and Entrepreneurship, and Director of Ontario Quebec Center for Organ-on-a-Chip Engineering. She obtained her bachelor degree from McMaster University and PhD from MIT. She's a fellow of the Royal Society of Canada, Academy of Science, Canadian Academy of Engineering, the American Institute for Medical and Biological Engineering, and Tissue Engineering and Regenerative Medicine Society. She received numerous awards and fellowships, including MIT Technology Review Top 35 Innovators Under 35. She was a recipient of the Professional Engineers Ontario Young Engineer Medal in 2011. Engineers Canada Young Engineer Achievement Award in 2012, Queen Elizabeth II Diamond Jubilee Medal in 2013, NSERC EWR Stacy Fellowship in 2014, YWC Toronto Woman of Distinction Award in 2018, and OPIA Research and Development Medal in 2019, to name just a few. So her research focuses on organ-on-a-chip engineering and development of new biomaterials that promote healing and attenuate scarring. She developed new method to mature human-induced pluripotent stem cell-derived cardiac tissue using electrical stimulation. Currently, she holds research funding from CIHR, NSERC, CFI, ORF, NIH, and the Heart and Stroke Foundation. She is an associate editor of the ACS Biomaterials and Science and Engineering, a member of the editorial board of Tissue Engineering, Advanced Drug Delivery, Reviews, Regenerative Biomaterials, Advanced Biosystem, Journal of Molecular and Cellular Cardiology, and eLife. She served on review panels for Canadian Institute of Health Research and National Institute of Health in the US. She's actively involved with BMES, Cardiovascular Tract Chair in 2013 and 14, and Termis AM, Council Member, Chair of the Membership Committee. She was a co-organizer of a 2017 Keystone Symposium on engineered cells and tissues as platform for discovery and therapy. She served on the board of directors for Ontario Society of Professional Engineers and currently serves on the board of directors of Canadian Biomaterials Society. Her research findings were presented in over 200 research papers, reviews, and book chapters with H index of 60 and over 12,000 citations. Her publications appear in prestigious journals such as Cell, Nature Materials, Nature Methods, Nature Protocols, Nature Communications, PNAS, among others. In 2014, she co-founded an award-winning company, Tara Biosystems, that uses mature human-engineered heart tissues in drug development. Tara currently tests drugs for major pharmaceutical companies. In 2017, she founded Qtero, a company focused on disrupting the skin regeneration and wound healing market through the use of proprietary Q-gel to promote scar-free wound healing. And this list goes on and on. And we are very excited to have Professor Radisic with us. Thank you so much. The floor is yours. And if you go on your video, you have the button for sharing your screen. Yeah.
Thank you very much, Human. It's a pleasure to be here. And here is my presentation. I hope that everybody can see that. So today I'm going to talk to you about the work from our lab that's focused on instructive biomaterials for healing and regeneration. If we think about regenerating...
Sorry, I interrupt you. There is a problem with sharing your slides. The slides are larger than the screen. Am I right, Mohsen? Is it true?
Yes. So we only see part of your slides.
Yeah. It's larger than the screen.
How about this?
This one, yes.
There you go. It works.
Yes.
Is it good?
Yes. Thank you so much. Yes.
Okay. So if we think about regenerating any organ, such as the heart, there are several things we have to consider. First of all, what are the cell types that make up this organ? And in the heart, you can see that there are many very highly densely packed cardiomyocytes surrounding extremely rich vasculature. And these cardiomyocytes are supported by cells such as fibroblasts, macrophages, smooth muscle cells, etc., and even neurons that are now proving to be extremely important for heart function. In addition to that, the vasculature is so dense that if you make the cast of the coronary vasculature, you will almost get the shape of the heart. And not only that, but there is a very remarkable structural organization of cells in the native heart. They're all aligned in parallel, And at the micrometer scale, they have very precisely oriented microstructure that is made up of sarcomeres. And then the contraction of these sarcomeres leads to tissue contraction, which lead to ventricle contraction, which moves the blood forward. So for us, when we're trying to think about how can we construct biomaterials to support cardiac regeneration, we really need to think about this structure-function relationship, that for us to achieve appropriate function, we have to have appropriate structure. The better function follows better form. And this is the overarching hypothesis of the work in our lab that is true for both work in organ or chip engineering as well as biomaterials for cardiac regeneration. If we think about what are the requirements for biomaterial design for the heart, it has to be elastomeric to support beating of cardiac tissue, and it also has to provide the certain alignment cues, and it has to be able to support multiple cell types. Ideally, also upon in vivo implantation, it should not induce any foreign body reaction. In addition to that, we want this elastomeric structure to be stable enough to enable handling of the material. If the material is too soft, we won't be able to handle it and implant it. And about 10 years ago, we came across this wonderful paper that Jian Yang published in Soft Matter, and it describes synthesis of POMAC using 1,8-octanediol, citric acid, and maleic anhydride through a very simple one-step single pot synthesis. And this reaction leads to a polymer that can be elastomeric and that cross-links both in response to UV exposure and in response to heat. We also found that we can make the mechanical properties of this polymer tunable if we add porogen such as PEG-DM, and addition of this porogen enables us to also control porosity of the material, to increase the mass transport properties, and to induce presence of these nanopores that you can see here that enable proteins and larger molecules to pass through the material freely. And I should say that basically reading this paper was really transformative for a number of projects in our lab in the area of organ-on-a-chip engineering as well as biomaterial engineering. And later on, we found out that we can really start to change monomers in this polymer and tune the monomer ratio, UV exposure energy, and porogen concentration to address a huge range of mechanical properties that are actually relevant for soft tissue engineering as well as cardiovascular tissue engineering. This work enabled us to develop POMAC polymer, enabled us to develop injectable tissues. And if we think about cardiac tissue engineering, when we want to place a patch on the heart, this is probably the most invasive of all procedures that you can imagine, because thoracotomy is required, where a chest would be open to expose the heart. And then the surgeon would place the patch on the heart of the patient, and in some cases, this was actually done in clinical trials in France, or they would, in animal models, apply the same. And we argue that minimally invasive surgery is really a better option. And that in particular, if we can have keyhole surgery, then we would be able to save the trouble of open heart surgery and make the procedure much safer. And so using POMAC polymer, we were able to design these injectable tissues for minimally invasive delivery. And so the shape memory is actually based on the design of the scaffold microstructure. In particular, we can see here that the scaffold can be injected in PBS bath. It opens up after injection. The scaffold supports beating of the tissue, and it also supports injection of the tissue, as you can see in the last video. Using the P200 pipette, we are able to actually inject this tissue. And the video is just running too slowly in a 6-well plate. The shape memory really depends on anisotropic properties of the lattice structure. So POMAC material by itself, it has elastomeric properties, and they come from the linear cross-linking, from the cross-linking of the linear chains. Basically, a 3D network is created in the polymer, as I showed in the couple of previous slides. And we, in the clean room, a super talented PhD student from my lab, Miles Montgomery, who's graduated since, designed PDMS molds that enabled us, through capillary action, to perfuse these molds with POMAC polymer and cross-link it with UV illumination. The molds afforded different lattice shape to the scaffold, and you can see that he graded percent of successful injection trials, as you can see in these videos, and percent of successful opening. So the video is illustrating what it looks like when the scaffold successfully opens and when we can unsuccessfully opens. And from these, we found that, for example, design 6, as well as designs 1 and 2, exhibited 100% of success in both of these criteria. But we decided to focus on scaffold design number 6 because these straight lines and the channel that was patterned on top of the straight scaffold lattice enabled us to control cell orientation. And so you can see what this looks like before and after injection. POMAC exhibits autofluorescence that is rather strong in green and red channel. And to us, that's actually an advantage because we can visualize the scaffold very easily. And you can see here that there is a live tissue around this POMAC lattice. And this tissue is alive both before and after injection, as evidenced by a very strong green color. In addition to that, the number of viable cells, the percentage really didn't change, and electrical excitability parameters such as excitation threshold and maximum capture rate that tells how excitable this cardiac tissue are remained unchanged. This is really important because it tells us that in the short term, the injection does not affect tissue function and tissue properties. It was also important for us to check if, in any case, the injection and passing of the tissue through a small orifice affects in any way cell death in a longer term. So what Miles did is he transfected cardiomyocytes with luciferase, and he used the syngeneic recipient to isolate cardiomyocytes. So this is all done in Lewis rats with Lewis cardiomyocytes derived from neonatal pups. And so if you look at injected tissue on the right and surgical implantation on the left, you can see that there is no difference in the bioluminescence signal over a period of 14 days, which tells us that really there wasn't appreciable damage to the cells done initially that could affect their function later on. In this particular case, surgical implantation under the skin involves cutting the skin flap and placing the scaffold, nicely opening and lining it under the skin and suturing around it. Whereas injection just involves pinching the skin, injecting some PBS first, and then injecting the scaffold under the skin in this puddle of PBS. Also, using micro CT of a scaffold that was labeled, we can see that the scaffold does indeed fully open up after injection, as these two areas exhibited some, although rather small differences in area of surgical implantation versus injected. The next important question was, does this biomaterial with cells improve functionally the function of the heart after myocardial infarction? In collaboration with the lab of Dr. Renke Lee at Toronto General Hospital, we were able to place this patch on, and we demonstrated that the infarct wall thickness was increased upon patch implantation. Ejection fraction was also increased, and basically diastolic properties such as relaxation was also improved, tau for relaxation time constant. So this tells us that the patch, through its own biomechanical properties, was able to improve function of the infarcted heart without inhibiting its relaxation. In collaboration with Chris Calderon, who was at that time at Sick Kids Hospital, we were able to place these patches on the surface of the pig hearts. And you can demonstrate, what we can see here, very profound beating of the pig heart. In a porcine model, the movement is a lot stronger than in a rat model, and this leaves the surgeon with very little space to work with. And now you can see here that there is a delivery port that is delivering the scaffold with the cells. However, we also noticed that if we add flaps and we really make the scaffold look like a Band-Aid, this delivery of the tissue becomes much better and easier for the surgeon to manipulate it on the surface. It's also possible to show this more clearly in delivery of the heart post vivo, and as well to look at this heart during the actual delivery. We can look at the heart post delivery. The patches are still there, and it's possible also to deliver multiple patches on top of the surface of the liver. Patches remain affixed to the epicardium after the heart is explanted. And just to illustrate some different locations a little bit better, you can see that it is possible to deliver a liver patch on top of the liver. The abdomen of the pig had to be inflated so that we have enough space for the 3 ports. One of them is basically for the camera. One of them is for the instrument that enables us to pull the scaffold, and one of them is actually for the scaffold. It was also similarly possible to deliver a smooth muscle cell patch around the aorta, as you can see here in this particular image. So this procedure and the patch is quite versatile. It can be used to access many different tissues. Importantly, in these single layer tissues, polymer processing allows us to also create modular structures. And using the same POMAC material and by adding hooks and loops to one layer of the scaffold, we are able to create modular tissues that we can build in the Z direction. And Miles published this work in Science Advances a few years ago. And you can see that this is a process that occurs in 2 steps. First, the basis with these posts is patterned. And then in the last step, the hook is actually placed on top. And you can see dimensions of this mesh here. And so these tissues are quite stable, these hook-in tissues. They are basically connected once the hook catches the loop. So in SEM images, you can see basically these hooks and loops. Once the hook catches the loop, the tissue becomes quite stable. It's stable with handling. And we do need to apply appreciable level of force to detach the 2 layers of the tissue. This particular patterning allows us to easily create mosaic tissues, and you can see we can arrange them in checkerboard patterns. These are actual labeled cells. We can have offset to the tissues for length extension, and we can also have angled assembly, which could potentially be important to mimic the alignment of the fibers in the ventricular wall that changes as we move through the ventricular wall. On-demand tissue disassembly is possible, and I would argue that this is one of the strengths of the current technique, because we can put 2 pieces of tissue together and very rapidly disassemble it without in any way affecting cell viability, and each of the single layers of the disassembled tissue remains functional, as you can see in this particular video. All of these patches that I showed you that were implanted on the hearts or that were stacked through hook-in tissue approach did not contain vasculature. And as I said in the opening slide, vascularization is extremely important for viability of heart tissues. It turns out that these elastomeric polymers are also able to help us to construct very versatile vasculature. Boyang Zhang from my lab, who's now a professor at McMaster University, came up with this concept of constructing angiochip, which is basically a polymeric scaffold, microfluidic polymeric scaffold that contains branching vasculature in the X, Y, and Z direction. Boyang elegantly creates this branching vasculature by 3D stamping, and he has published a detailed protocol that should enable other labs to do it, hopefully in Nature Protocols. And what you can see here is that he starts with standard SU8 molds, and then he creates layers of angiochip scaffold through capping the PDMS mold on the glass. And then he perfuses the POMAC polymer. He cross-links the polymer with UV illumination, and then he removes the mold. And upon cross-linking, the polymer will remain bound to the glass. And then he can build that up in the Z direction using the next layer. And because POMAC likes POMAC better than PDMS, it will actually, after cross-linking, remain stably cross-linked. The layers will remain stably cross-linked. And this enables you to build the structure in the Z direction. So we have a pretty good control of building the structure in both X, Y, and Z direction. And you can do this by ordering also these SU8 silicone molds if you don't have access to the clean room. All you need is a crosslinker, an XY UV crosslinker, and XYZ stage. So the process is manual, but it does enable you to create multiple angiochip scaffolds in parallel, as you can see here. This work enabled us to create the vasculature that is both mechanically stable for handling and permeable, because polymers usually suffer from this limitation that you cannot really control their permeability very well. So what Boyang has done here, he incorporated two levels of permeability, one through these nanopores and the other one through microholes. And the microholes also enable connection of endothelium to the surrounding space where the tissues are located. He builds custom-made bioreactors that have that are feeding these tissues by perfusion or gravity-driven flow. So what you can see here is that there is one well upstream of the tissue, and this particular well will be your media reservoir well, and you can fill that with more culture media. And then there is less culture media in the downstream well, and these tissues will go right in the middle. The first step, you can see one angiochip tissue in this custom-made bioreactor. The first step in this procedure is endothelializing the channel lumens. The second, and that usually takes about 24 to 48 hours. And you can inject basically the endothelial cell suspension from both sides of the lumen, and they will be drawn into the lumen, into this branching vasculature by capillary actions. And so if you wait for 24 to 48 hours under gentle rocking, you will be able to endothelialize fully this scaffold. The next step is sealing of the cells using a hydrogel around the scaffold, and then if you wait for several days, the process that includes this cell gel compaction will lead to formation of stable tissues. And you can see that, I should also mention that these polymers here are usually gelatin coated to further support cell adhesion. And you can see that it is possible to create nicely confluent endothelium on these inner lumens, and that even the corners are well covered. Through the 3D stamping technique, one really nice feature is that we can control apparent mechanical properties of this entire scaffold. So we really do have multiple levels of control of tissue properties. The first level of control is through polymer chemistry. And then we create material that has tunable properties. Then we could go from 50 kilopascal of real Young's modulus to over 1500 kilopascal. But in addition to that, by tuning the lattice shape, we can tune apparent mechanical properties of this a scaffold. So these are not true, this is not a true Young's modulus because of the holes, but it is basically a parent Young's modulus. And this enables you then to control anisotropy. So for example, anisotropy or mechanical properties is very important for cardiac tissue because in the long fiber direction, the tissue will be much stronger than in circumferential fiber direction. And so we will be, through this angiochip design in scaffold B, we are actually, if you look at these numbers in B and you compare that to adult right myocardium, we are able to mimic the mechanical properties of the actual myocardium. And you can do the same thing for liver or any other tissue that you are interested in mimicking. Once you see the cells, they will go all around the scaffold struts, and all of the pores in the scaffold are interconnected in the Z direction. And this now enables us to create a tissue that twitches and that propagates electrical impulses. In addition to that, we can implant this tissue by direct anastomosis. You can see the inlet and outlet here. And we'll just hopefully let that move around. And we can establish flow immediately. So this was really one of the first, probably the first polymeric system that doesn't include the use of autologous vasculature, such as that in flap surgery, that enables you to hook in the plumbing, hook in the vasculature directly and establish the flow directly in the scaffold and immediately upon implantation. And you can see that we have artery bypass configuration and artery to vein configuration that we can use the scaffold in both settings. The tissue in this particular case remained implanted for 1 week, and you can see very nice tissue integration at the time of explantation. You can see that these, if we use surgical anastomosis, these endothelial cell channels will remain open. In some cases, we did have some thrombosis, so it's not perfect. And we can see in many channels that these endothelial cells really remained on the scaffold wall, but that's really for 1 week after implantation. We can also see infiltration of smooth muscle actin-positive cells, survival of implanted cells, as demonstrated by troponin T staining. Once you work with these polymers, it is actually quite possible to now try to model some of their other properties, such as we could incorporate carbon nanotubes to increase polymer conductivity, because the polymers are well known for not being conductive. And conductivity is important for cardiac tissue because the electrical signal travels mostly through cardiomyocytes, but also around them. And so it is important to have an environment that is conductive. And so what Alok David Porthire from my lab did, he was also extremely talented postdoc and a graduate student who drove the development of new polyesters in my lab. You can see that once we incorporate these carbon nanotubes, the tissue beats much better, much stronger force of contraction. Also, electrical excitability parameters such as excitation threshold are significantly improved. We can see a lot lower excitation threshold, which is better in this particular application. In addition to that, Locke was able to now start replacing specific monomers to enhance polymer elasticity through chemistry. So for us, polymer elasticity is important because we're working with elastic tissues, and having a 3-carboxylic acid is important for the ability to create cross-link chains, because it's through this cross-linking that we actually get elastic polymer properties. So when Locke replaced citric acid with this 1,2,4-butane 3-carboxylic acid in what the same chemistry that was used to make POMAC, he was able to create a new polymer that had improved elasticity compared to POMAC. And it was actually, we were able to stretch it much more with the same stress. If you can look, for example, here. And we are also able to grow very nicely oriented cardiac tissues that have well-developed cross striations and that exhibit staining for cardiac gap junction proteins such as connexin 43. In addition to that, another good candidate is itaconate or itaconic acid. And to further create more versatile polymers, Locke now used a two-step procedure to create these polymers that we called PICO, that contain both itaconic acid and citric acid in them. And by doing that, you can see here how we are able to create a range of tunable mechanical properties, going from extremely soft polymers to hard polymers of over 1500 kilopascals. Doing that also enables us to tune percent mass loss, which is important if we want to have tunable degradation in vitro, and in vivo as well. This work was published last year in Advanced Healthcare Materials, and you can see that these picopolymers enabled us to create beautiful cardiac tissues that are beating quite strongly and that contain aligned cardiomyocytes. Because picoelastomers cross-link much faster than our other elastomers, including those like POMAC or elastomers based on 1,2,4-butane carboxylic acid, Human, who is the organizer of this seminar series, was able to use these picoelastomers in a 3D printing application. So using extrusion printers, and if you use a coaxial print head with the carbomer as a sacrificial layer, Human was able to really rapidly print these tubular structures and use them to create some of our organ-on-a-chip platforms. In the other body of work from our lab, these organ-on-a-chip platforms are all created by 3D stamping, which is more laborious than the 3D printing procedure that Human developed here. So you can see these tubes that he's able to 3D print, and they do have a good level of microporosity that really increase the permeability of these materials. But once we started working with itaconic acid, and we really, when we were thinking about how do we increase the versatility of mechanical properties and also tune the degradation rate of these scaffolds, because we don't want them to hang around on the heart for a year or 2 years, we came across itaconic acid, which is a really wonderful molecule. It's a tricarboxylic acid that was discovered by Dr. Hans Krebs, but because it was really not so important in respiration, it was kind of forgotten until the beginning of the 21st century, when people discovered that it can tune host response. Basically, it tunes innate immunity. And we thought this would be an opportunity to start thinking about biomaterials that can solve some of these well-known problems in biomaterial science. And that is that synthetically derived biomaterials elicit a foreign body response. Some of them do, and this is very well known. So you can see hallmarks of foreign body response, such as the appearance of these giant cells, overproliferation of cells, the presence of dense extracellular matrix, and eventually fibrous capsule formation. And so Locke then set out to design a new polymer that will incorporate the itaconic acid and release it slowly. And itaconic acid is great because it gives us an opportunity to work with, create tunable chemistry, but it also has this reactive moiety right here that we want to maintain, and we don't want that moiety to react, because then we won't be able to release this dicarboxylic acid into the environment. And so what Locke then decided to do is to kind of develop the chemistry that will enable us to preserve itaconic acid and then later on measure if this polymer really exhibits hallmarks of immune regulation that was described by itaconic acid. And so basically, this describes the concept of polymer design that he used. And one of the main readouts was to assess its effect on macrophages in vitro and in vivo. The problem is when administered in soluble form, itaconic acid or itaconate have a very low half-life of less than 24 hours. And so the question is, does the biomaterial enable it to extend that delivery time and also activity of itaconic acid? And so what Locke did here is he created linear polyesters from itaconic acid and also 1,8-octanediol, and as well as other alcohols. And you can see here that if you control the alcohol chain length, you can create materials of different appearance. So you can have a liquid, you can have solid, or you can even have gel appearance. But it's still a liquid, it just appears as a gel. And so Locke was able to demonstrate successful incorporation of both itaconate and these specific alcohols into the polymer. And then he also was able to demonstrate that he has a slow and sustained release of itaconate from these different materials, that you can have accelerated degradation under, for example, alkaline conditions, such as those that are present in the peritoneum, and that you can also slightly accelerate this degradation if you use enzymes, such as lipase. However, bulk material properties of these new polymers remain stable over 1 month, indicating potential long-term stability. What Locke did next is he did a series of experiments with activated bone marrow-derived macrophages to demonstrate immune modulation. And what we are looking for here is basically ability to attenuate inflammatory response of macrophages. So for that, we first activate macrophages with LPS and interferon gamma, and then we measure how much different cytokines they secrete on our itaconate-containing materials or representative controls. So one representative control is widely used polyesters, such as PLGA, and you can see that in a number of these important cytokines, itaconate-containing materials, as well as soluble itaconate, really downregulated secretion on inflammatory molecules in these activated macrophages. And we can see very familiar, strong response on PLGA, basically in all of these materials. In addition to that, Locke was able to measure reduced nitrate content, which was also one of the hallmarks of ITA, soluble ITA presence in the body. And basically, this molecule is normally secreted by macrophages in the body to control inflammatory response, as well as to participate in bacterial and viral clearance. And as a part of its immune modulation, it does exhibit this reduced nitrate content. What's really important for us to demonstrate is that we can capture some of the bacteria killing properties of itaconate. In the body, macrophages will secrete itaconate to kill off bacteria. But this only works under the conditions of limited kind of substrate availability. And itaconate works by inhibiting glycosylate-based growth of bacteria. So in the body. So what Locke was able to capture here with these polymers as well, outside of the body in vitro settings, is that we can inhibit growth of E. coli on acetate, which really shows that we have mechanism-specific inhibition of growth. And this is done by itaconate and not by side effects of other polymers. And you can see that really if we compare under those conditions itaconate to silver, we can see very similar levels of inhibition. So that what the polymer is releasing is really a functional form of itaconate. In addition to that, we can see that our controls such as PLA or even PLGA, these additional controls that we did that are not shown here, do not exhibit any kind of bacterial growth inhibition. However, if we use non-inhibitory glucose source of carbon, then we don't see any differences, which still, again, tells us that these effects are due to itaconate. Because we can have very viscous liquids that look like gels, that technically not gels from the rheological point of view, but they look and feel like gels. They are viscous liquids. We can have also solids, as well as less viscous liquids. It is possible to coat these materials on various surfaces, such as tubes or implants. So you can see coating on tubes, you can see coating here on titanium implants, and it is also possible to create discs of polymer and scaffolds of polymer for cell culture. And so you can see here that although itaconate that's released from this material is inhibitory to growth of E. coli, it is not inhibitory to growth of fibroblasts and mammalian cells at all. So you can see cells grown of itaconate-containing polymers versus those grown on tissue culture polystyrene are exactly the same in terms of cell viability. But really the final and the most important question here with the view of performing immunomodulation in vivo is what is the response of cells in the body to this polymer? So there are simple questions here that also pertain to toxicity and biocompatibility, as well as ability to control innate immune response in the body. So what Locke did is, using a murine model, he injected this material into peritoneal cavity of mice. So what he then used as a control is silicon. And the reason he did that is because of comparable mechanical properties between viscous liquids, which are these itaconic acids containing polymers, and silicon, which is very widely used in different implants. And then at 2 time points, he measured the presence of neutrophils, early monocytes, and eosinophils in the peritoneal cavity. And this is possible by rinsing and harvesting all cells from peritoneal cavity. And as you can see here, that in all of these cell types, with time, there is a very rapid resolution of inflammatory response with itaconate, and the inflammatory response persists with silicon. And basically, looking back at these data, it is really not possible to prevent inflammatory response from occurring fully. But what we want to see in a biomaterial, that it does resolve quickly and that it does not persist for a long time. And through Locke's work here, we demonstrate that itaconate-containing polymers are capable of giving us that functionality. Another important thing that we can notice here is that if you have any intervention to the body, such as surgery or injection, it is very well known that you will fully deplete tissue-resident macrophages. These tissue-resident macrophages are different from bone marrow-derived macrophages, and they're very much implicated in healing. So in addition to resolution of inflammation, you want to have a rapid return of resident macrophages. And you can see here that with itaconic acid, we have much faster return of resident macrophages, albeit not to the uninjured levels compared to silicone. That still basically doesn't have any appreciable level of resident macrophages 10 days after the injection. So through this itaconate work, we saw that biomimetic bioactive polyesters can deliver ITA through hydrolytic degradation release. And this really harnesses the opportunity of these biodegradable materials to deliver immunomodulatory molecules. And we saw through a number of tests that are focused on confirmation of what is known about the effect of ITA in the body, that we can regulate both inflammation and infection in a very specific manner that is consistent with the ITA mechanism. And so in translation, that may hold value in basically constructing new biomaterial scaffolds from these polyesters that will enable us to accelerate resolution of inflammatory cell infiltrate and to some extent prevent implant infection. And it's important to note these are not antibiotics and they cannot systemically eradicate bacteria, but under the a limited glucose source, such as that found in biofilm or surgical site infection, these biomaterials could push that balance towards resolution of, have that potential to push the balance towards resolution of infection. And so basically, in the interest of time, I'll just skip to the next slide and I will summarize that I showed you several important technologies from our lab, and those include injectable tissues that are all demonstrated here, angiochip, and 3D stamping for creation of hook-in tissue. So that's a family of technologies that we developed with POMAC. And then by starting to change monomers in what was essentially chemistry that was described for POMAC by adding another tricarboxylic acid, or by adding a dicarboxylic acid such as itaconate, we are able to further tune elasticity of these polymers, as well as to tune degradation rate and to basically tune the host response of the body to the polymer implantation. At the end, I'd like to thank everybody who helped with this work. There's people in my lab, our collaborators, and our funding sources, and I'll be happy to take a couple of questions right now. Thank you very much.
Thank you so much, Professor Radisic. Mohsen, please go ahead.
Well, thank you very much, Dr. Radisic. It's always good to hear, to listen to your talk. I haven't heard this talk before. It's very interesting and very exciting. And I envy, when I look at your background on your wall, I see many, many frames and awards and cover images that catches my eye. So, well, congratulations on so many achievements that you have.
Mohsen, you only see one side now.
Yeah.
It's all around the office.
So I don't know how much time do you have, Dr. Radisic? You told us that you have another meeting coming.
Yeah, I have another meeting at 1, but I do have time for like 5 minutes or so for questions. So,
oh, wow, okay.
Thank you very much, Mohsen. I wanted to make this talk because of Human. He invited me, and he was in my lab working on these polymers. And I know I usually give organ-on-a-chip talk, but there is a big body of work also on polymers, lots of exciting stuff that Human and Locke and Miles did over the years. And so this is an opportunity to showcase that work as well.
It was amazing. I really enjoyed the talk, and I learned a lot from some of the, especially the technologies that you developed. There are many questions for you, but I'll just pick the first few questions, and then I can share them with you, and then maybe later on we can provide more feedback to those who ask the question. So we'll start with the first question from Xavier. How do you promote adhesion of the implant to the tissue?
So Javier, we used in all of the implantations that you saw on the pig, we used fibrin glue to glue the scaffold on. In the red heart, it was just sutured. But my student, PhD student, Dawn Bannerman, she just had a paper that was published in Advanced Biosystems on the cover, literally a few days ago or a couple of weeks ago, that describes how you could modify a patch with dopamine, some of these polyesters with dopamine, and you really create something that looks like a Band-Aid that is adhesive on two ends, and in the middle, it's not totally adhesive because you want the tissue to grow there, right? So you have that dual functionality. You use these flaps to kind of glue the tissue on, and in the middle you're growing the cells.
Wonderful. The next question is from Didarul. I hope I pronounced your name correctly. Could you comment on challenges in storage and transportation of these tissue patches?
So if you don't have cells, if it's just a scaffold, there is really no challenge in storage or transportation. These are pretty stable materials and they could be available off the shelf. However, if you want to grow the tissue somewhere and then ship it somewhere else, obviously it needs to be done under GMP conditions in a closed system. And my lab doesn't have the bioreactor that satisfies those criteria. And I think it's really the industry that will drive development of these, because it's very, very rigorous and time-consuming work. In GMP, most of it is documenting that you didn't do something wrong, right? And so I think that's the opportunity for to advance new bioreactor systems.
Awesome. So I'll ask you the next question, and then again, I mean, when you need to leave, just let us know, because there are many questions. 17 questions in line. So,
okay.
So what is the degradation rate? Oh, that's from Courtney Hosley from McGill. What is the degradation rate of the ITA materials? And then the second question is this degradation rate depends on the implantation site?
Yes, totally, yes. So let me just find the slide. It does depend on the implantation site. That's why you will be able to tune it. So the different parts of your body have different pH. And I'm just looking, where is that slide that shows the? It's right here. So I should be able to share it. Under alkaline conditions, for example, we found that it degrades really quickly, and we did the alkaline conditions because you can see here, you can see how it drops like really quickly over just a few days under alkaline conditions. In 7 days, it will degrade almost fully. And if it's just hydrolytic degradation, then it will be much slower, and it will also go a bit faster under acidic conditions. So it really depends on where you're putting it in the body. And but I think having these polymers also gives you that ability to tune.
Wonderful. The other question is again about the ITA material. Does ITA play a role in promoting vascularization? That's from Rohin.
Oh, Rohin, great question. We don't know yet. We'll have to do more studies in a classical sort of subcutaneous implantation to study that, because the peritoneal model that Locke used, we just collect all of the cells from peritoneum, and we can't really tell where the biomaterial is, but we know it's there. So we didn't do that kind of characterization yet. But it's a great question for next steps.
Okay, wonderful. The other question was from Garima, but I think it's the same as Hosley's questions. Are these ITA material site specific for their desired action? I think you already responded.
Yeah, I think we can say that they are, because it really depends on pH and in different sites in the body.
Okay. Are we still good with a couple of questions? Or?
Yeah, let's take two more and then I'll drop off.
All right. So the next question is about the autofluorescence of the scaffold. So that's from Bisan. How did you differentiate between autofluorescence of the scaffold and the dead cells?
POMAC is, so we will look in between, right? So we will look in between if you look here, I'll share my screen from Mars's work. If you look in between the scaffold pores, then that's the area where we focus on. We will not look on the scaffold strut to characterize this cell viability. Maybe I can just leave okay. So if you look here, right, if you just look in between, you can see that it's all green. So that's what we did. And we don't measure on the actual scaffold strut. But I should also inform you that PICO is lot less autofluorescent, lot less. And so we now came with, like, I think that's a good property not to have. Like, you don't want to have autofluorescence that's so strong in injectable tissues or in scaffolds, and PICO is a good, really good candidate if you don't want to have autofluorescence. However, we love autofluorescence of POMAC in organ on a chip applications because it allows us to measure force of contraction in our INVADE platform and in BioWire platform just by looking at the movement of cantilevers and wires.
We're talking about the cell paper, I think.
Yeah.
Great.
Yeah, I can take one more question then, and
then we're good. Okay, so just the last question is from Dida Ru. So were these injectable scaffolds seeded with cells before implantation? If so,
yes.
cell type was used?
Yes, they were, and they were grown for at least 1 week. You have to really make sure that the tissue is stable, and we seeded that. We made different types of tissues. We made cardiomyocytes, so cardiac tissue. We seed with cardiomyocytes and fibroblasts. We seed with liver tissue, with liver hepatocytes, and we seeded with smooth muscle cells. There are two things that are really important for this. And so we published a protocol on how to create these injectable tissues in ACS, Biomaterials Science and Engineering, I think in either 2017 or 18, and the lead author is Montgomery also, if you want to see like more detail. Two things are really important. So you need to have a lot of cells, and we always spike in a little bit of fibroblasts so that you get that nice gel compaction. And you have to make sure that the cells go all around the scaffold, not just on one side. So we have a bioreactor that lifts the tissue off the bottom of the bioreactor a little bit. It's a microfabricated bioreactor, and the cells are going all around. If they're only on one side, it will just slough off when you do the injection. The cells won't stay. Okay.
Thank you so much. Thank you so much, Dr. Radisic. It's always, great to listen to your talk and learn about different technologies. Dr. Radisic is not only like a great scientist and pioneering scientist, she's also a leader and a great educator. So we had a problem of transport phenomena. She with the pen, started, teaching me, how to solve it. So I have your handwriting, so I always.
Yeah.
So thank you so much for your time, and we had a great turnout, and then I think also audience benefited a lot from the technology that you developed in your lab. And by that, Mohsen, do you have anything to add?
No, I have many questions, but I mean, in the interest of time, I think we can end the lecture today. This talk. Thank you very much again, Dr. Radisic, for participating.
I just want to remind everybody that we have Professor May Griffith from University of Montreal next week, same time, same place. And we would like to thank also our sponsors, Transmedtech Institute and Fluid Foam Company. So see you all next week, Wednesday at noon today, same time, same place. Thank you so much, Mohsen, and thank you, everybody.
Bye.
Bye-bye.